Chapter 8. You don't have the best of me. You're not the boss of me. We are going to finish you this video. So, we have lots of information about enzymes. Oh, so much. This is what we're left with. Last one, 8.5. And I kind of feel like, hey now, I think I talked about that. What more could I possibly say? So, let's just go back here. You said, describe, so I need to make it to where I have like this picture in my head and tell you why it matters. Allosteric, I know you. You're that weird site that's not the active site in an enzyme. Regulation, I get that. Control of the enzyme. And be sure you talk about activation and inhibition about around that site. I think I did that. Did I not? Let's go back and look. So here we're talking about using some type of molecule that's going to sit here in this allosteric site. When it does sit here, all of a sudden your enzyme is not in the right configuration. The active site changes its shape. And so now the substrate can't bind, and you're not going to get the complex, and you're not going to actually make a product. What more could you want from me? Oh, more. So, turns out that little, I don't really have a name of my own. I mean, it's almost like, okay, so if your parents had your brother, and they named him Tom, and then they had you, and they didn't name you like your own name, they named you not Tom. That kind of would make me feel bad. That's the way I feel about the allosteric site. Active site, you got a name. Allosteric site, you're not active site. Well, that's kind of crappy. It's really important. Honestly, if you think about it, so on number 1, this allosteric site is kind of like a volume knob. Because if you think about a volume knob, you can turn something up, you can turn it down, and if you go click, you turn it off, right? That's exactly what this is like. So we're going to think about this. Allosteric, hello, you're not the active site. Site, regulation, works much like a volume knob. That means that, yes, I've talked about using the knob to turn the enzyme off, right? But I haven't talked about using this site to turn it up. How would that work anyway? That's this part right here with activation. So, some allosterically regulated enzymes. That's a mouthful. I'm just going to write it. Some allosterically regulated enzymes. So ones that use this volume knob-like property of this allosteric site, they are actually going to be activated through this allosteric site. So some of these that are regulated through the allosteric site, these enzymes are activated, turned on, turned up, turned on, turned up, which means works even better, by that allosteric site. Okay, so how do I want to put that? So we'll put binding of a regulator molecule to the allosteric site. So what this means is, for those molecules, so I'm going to draw a different enzyme so we don't get confused. I think he's still going to be brown. I'm going to change his shape. I'm going to draw the one I always draw before. It's kind of sad looking. It looks like Pac-Man. It's like my favorite way to draw. So this is my enzyme. This is my waka 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 active site. Over here is my allosteric site. How do I know that? Because it's a site that's not the active site. That's how, not Tom. So this is my enzyme. This is the active site. That's where the substrate would bind. That's where the actual catalyzing of the reaction occurs. This is the not active site. So this is the allosteric site. And this whole enzyme in this configuration, an empty allosteric site, is off. So this enzyme is off. So, seriously? Let's get a different one. What color did we make the substrate? Green? So, I'm going to have a substrate. This is substrate. And cannot bind. Because this particular type of enzyme is regulated allosterically. So now let's turn it on. So now I'm going to draw it where that, I'll start over on this back end of Pac-Man. So there's the allosteric site. Let's put whatever this regulatory molecule is. So now the allosteric site has been bound. So this is going to be some molecule that regulates the enzyme binding to the allosteric site. Now, the binding here changes the shape of the active site, which I drew this way on purpose. I'm not the best drawer, but you can definitely tell this active site is way bigger than the substrate that's supposed to fit in there. So once you have this regulatory molecule bind to the allosteric site, your active site changes shape. This is still my enzyme. My enzyme now is on. The active site has changed shape, so the active site is now the correct shape, and my substrate molecule can go in there, can bind. So the substrate fits. Now we're working. We work as long as this molecule hangs around. If it leaves, we go back to this shape and we no longer work. This type of regulation is really, really important. We're going to talk about it here, but this is a super duper smart way to turn the lawnmower and the blow dryer off. Because otherwise, if you don't have some type of regulation, that enzyme is working nonstop. And we don't want that. We need some type of control. So this is the way this works. So we've already talked about inhibition. So we know that thinking thou. Lord, I'm talking crazy. So let's go to okay. Number 5, I'm just going to write here for inhibition C 8.4, number 5, non-competitive inhibition. Because we've already talked about it. But what we didn't talk about was, well, I can also use that site, that allosteric site, as a way to turn on the enzyme. Sometimes you have different molecules here, so let's put that in there, because it did, I mean, if my analogy is the volume knob, it's not just on or off, right? It's on or off or more. So some molecules, so you might have, so this one fits. You could have a different molecule. So molecule 2, and that it regulates the enzyme by binding to that allosteric site. But just like we learned here, where, you know, a little bit of change in shape makes it a little less effective, maybe this one's even better than the purple one. Maybe it makes this shape of the active site the best. So that would optimize the rate. So you would have the ability of having to fine-tune or tweak the shape of this active site, which means you fine-tune or shape the function of the active site. So if this is a better fit, then you would have a better structure, so a better active site shape, right? Which would equal better ES complex, fits better, which is an increased rate for the enzyme. Kind of cool. It lets you tweak this activity of the enzyme. I fit okay, you fit okay, we work okay. Ooh, I fit best, you shape yourself best, we work best. I'm not here at all, your shape's wrong, we don't work. So that's allosteric regulation. Holy cow, I don't know what to put on that to tell you how important that is, because it's a huge big deal. We're going to talk about it nonstop because of this part. Okay, so we finished allosteric regulation. Super important. And the reason why it's important is because we need to think about, with question number 2, what feedback inhibition is. So let's just picture an assembly line, and you turned it on, and you're going to continue to make whatever the product is, and you don't have any idea, you don't have any information of how much you've made, if it's enough, if it's too little, you're just making it. It's extremely inefficient, and it's not going to be the way that cellular processes work. So let's just take these words. I think what I'm going to do is just remind you again, we're talking about feedback inhibition, and then we want to explain how we use that to control cellular products. All right, so this is 2. And I'm just going to put up here feedback inhibition. And I'm going to just translate this so that we don't get wrapped up in that's a biological term. Feedback is nothing more than information. So, for example, you turned in your intros to me for the lab with the pill bugs. I gave you feedback. I gave information about just ways that it could better align to the rubric. And then you used that information to make important, necessary changes. So, in this particular case, there's information that's going to be used to inhibit continuing to make product when you don't need it. So inhibition here is just self-regulation, just like we have inhibitions, and they're good for us. They keep us from doing things we shouldn't. So self-regulation. So in the context of biology, what they're talking about is this. If you have a pathway and you're going to start off with some molecule, and we'll call it 1. And in this pathway just means you're going to have several reactions put together to end up with a product. So these are sequential. That's what a pathway is. So we'll put pathway over here, and it's just a series of sequential reactions. So we're going to have molecule 1. It's going to be acted upon by enzyme A, and in that process, as it is acted upon, it's going to be converted to molecule 2. Molecule 2 is going to be acted upon by enzyme B, and it will be converted to molecule 3. Because a lot of times, you can't go straight from one to the end in one step. There's a lot of bond changes that need to be made, and you can't just make them all at once. So, you know how people make the joke about how do you eat an elephant one bite at a time. So this is done stepwise, and it has to be done sequentially because there's an energy stability that's involved. So let's just keep going, see where we're going here. So enzyme C is going to convert 3 to 4. Enzyme D is going to convert 4 to 5. So ultimately, this pathway would be making of whatever this molecule 5 is, and we would have had 4 enzymatically catalyzed steps. So this is a pathway, and the concern is, so here's the problem. The problem is, as long as you have 1 and you have these enzymes, you will make 5. That's all there is to it. So how do you regulate how much product is being made? Much better. So if we want to think about it in other words, I want, when I have a low concentration of 5, I want the pathway to be on. I need more 5. When I have a high concentration of 5, I don't want to make any more. I have plenty. I want it to be off. How do you do that? The coolest, most beautiful, simple, efficient way is, most of the time, feedback inhibition is through regulating this particular product, what you're using, by inhibiting an enzyme at the very beginning. Because if A can't work, 1 can't go to 2, and the rest of it won't happen. So let's think about how that works. So you're going to often, because it makes the most sense, you're going to use the final product, in this case, which is 5, to shut down the pathway. So let's see what that looks like. So we'll go back to the enzyme that has the allosteric site. So I'm going to draw yet a different one. I'm going to draw this enzyme A. I'm going to give it an allosteric site. That's my on-off switch, my volume knob, my regulatory place. It is not where that we actually do the active site work. And I'm going to let that be, I don't know, 1, 2, 3. There we go. So this is somehow what my brain has decided. This is enzyme A. This wonky looking fist looking thing I've decided will be my active site, and here will be my allosteric site, my regulatory site. And so let's think about what goes where. 1 would go in the active site, right? So molecule 1 this is 1, would go in the active site. That's what A works on. It works on the first molecule in the pathway. Over here, we're going to use the allosteric site to turn A off. So right now, there's nothing there. Let's use this one. A is on. So we're going to make 1 to 2, right? Assuming that we're going all the way to 5 at some point when all the rest of it starts going. So I'm going to start making a lot of 5. So we're going to start putting, got a lot of 5 being made. This is my final product. At what point do I stop making 5? When you have this high concentration of 5, which that's what this condition is, I now have a high concentration of 5. What can happen is, so this is the on condition, you're going to make 1 because this enzyme is on. The rest of the pathway proceeds to completion. You make a bunch of your product, which we've called 5. I'd like to stop making 5 now, please. So what would happen to turn A off? The most brilliant, simple, beautiful way to do that. So here's A. Here's its substrate, which is 1, goes in the active site. This is A. I would like to turn you off, please. So the easiest way to do that is to let your actual product, which I have a lot of right now, be the inhibitor of A. That's where feedback inhibition comes from. feedbacks information. This information in the cell is there is so much 5 that not only can we use it for what we intended, but there's enough that it can sit in this allosteric site. So this is 5. This is your product. And that product, because it's in excess, now turns A off. So we are not going to make 1 to 2. That's been turned off. And what's going to happen over time? Well, we're not going from 1 to 2, which means 3, 4, 5 is not going to happen. You're going to get less and less concentration of 5. We're going to use it. So once we don't have very much 5 left, 5 will leave that allosteric site because it's going to be used for what it was intended for, which will turn A back on. So at this point, the next piece would be, once the concentration of your product is low, then you're going to have your product, which is 5, leave the allosteric site. It just doesn't have an affinity for it anymore. It's not attracted to it anymore. It's more attracted to whatever its job is to be done. So it's in short supply to be used. So it's going to leave the allosteric site. So therefore, A is now on again. So you would have the condition we had here, which is low 5. So 5 would leave, go be used, which means that your active site has the correct conformation now because 5 inhibited, so it made that active site not work. And so at this point, based on the information that we're low again, 5 comes out, A turns back on. And this goes back and forth, back and forth. So the summary of it, the take-home, the key, is the product itself is the on-off switch for the pathway that makes it. How does it do that? The way it does it is based on concentration of the product. The product will sit or leave the allosteric site. Allosteric site. Pretty beautiful. Pretty simple. You always block the first enzyme of the pathway. You wouldn't want to block B, because then you could freely make 2, and you don't want 2, you want 5. So you go all the way back to the beginning, that feedback, that information of how much product did you say we had? a lot. Wow. So much that we have plenty where we could sit in the allosteric site and turn this first enzyme off. Okay, that's what we'll do. So now we're off. How are things going? We're still good. Well, we're getting kind of low. Whoa. We're low. Well, okay. Out of here, I'll come be used. Oh, all of a sudden, the pathway, that assembly, now is on, and we're making 5 again. So when this happens, 1's going to be converted to 2, all the way to 5, and you'll start increasing your concentration of your product because your pathway's on. It's kind of neat. So think about what it has as the beauty of it. Because the product actually turns the pathway on and off, this feedback inhibition, this process of regulating using information in the form of concentration of the product, that's what this is. Let's reword that. So it's regulation of a pathway based on concentration of the product. Pretty brilliant. It's going to prevent the fact that you would overproduce, overproduction of the product, because the concentration of the product drives whether the pathway's on or not. So if you also think about this, this is what every company does. They have the ability to count the number of items they've created. And when they reach their quota, they stop making them. Well, cells can't count. So it seems like, well, bless their hearts, then I bet they just can't do that. Yes, they do. So even though cells can't count, so they can't make the point, we've met our quota, let's stop making that, what they do use a mechanical way to keep up with the amount of product. So concentration is just, you have a lot of it in an area. Well, you're going to have the product in the area where the pathway is, where it's being made. So all of these enzymes and these particular substrates that are there, they tend to all be in proximity to each other because that's more efficient. Just like, you know, when you go and you look at an assembly line, that's really what this is like. So we're going to think it's a cell, I can't forget to write in cursive, assembly line. That's all it is. And when you make a lot of product and it stays around here, there's going to be, through just molecular movement, what we've talked about, and through collisions, there'll be more collisions with 5 with that allosteric site. Because there's whole bunches of 5 right now, we've made a ton. That's this situation. So as you use it and your concentration decreases, you won't have collisions with 5, the product, with this site. In fact, this product will lose affinity for that enzyme because it's going to be drawn to where it needs to be used for work, and then you have the open site again turning the pathway back on. And then you'd start making 5 and you'd accumulate the product. So what I just said is it's all based on 2 things. Affinity, which just means attraction, between two molecules, locations. It's all based on attraction, and it's also based on, so that's one of them, and the number of collisions, which is based on concentration. The more you have, the more you have to collide. So, pretty cool. So one way you could draw this, I pay attention to this because this is one of the more difficult test questions on the test coming up. So if you think about this concentration of product, and we're going to just draw it out. So we're going to have, the pathway is going to begin, it's going to make product, so the pathway is on. Once you reach this maximum threshold, which is going to be different for everything, for the concentration of product, which means I have enough now. There's going to be also a very high, so this is going to be a high concentration of product. It's going to be a high number of collisions. So at this point, there's enough product and collisions for that product to sit in the allosteric site, and then what will happen at this point is the pathway turns off through that allosteric site being occupied by the product. You'll use the product, so the concentration is going down, going down, going down, until you get to where there's, we need more product now, a minimum threshold. Do you know what threshold means? It just means level of the concentration of the product. And we will have very few collisions because we just don't have much product. So at that point, the product will come out of the allosteric site. Enzyme A turns back on, and you'll have this cyclic process of on and off. A lot like a thermometer, like a thermostat. So you have the ability to turn it on and off based on concentration. Now, the next question, we said, wouldn't it be efficient if all of these enzymes were really associated with each other so that you're not running around moving 2 way over here because B's over here and then 3's being made and you're hunting for where C is? I don't know if you've ever had that happen, but when you're going somewhere and you have to go hunt for everything you own, like you don't know where your phone is, you don't know where your keys are, it makes the whole process very inefficient. So 3 states describe the purpose of localization. What that means is putting things in the same location. Of enzymes within the cell, include in your description the order of enzymes in the membrane. Well, we've learned an awful lot about proteins in the membrane, and part of what we learned with our whole, what type of proteins are there in the membrane? SIC-T would be, there are enzymes in the membrane. So if we have a membrane, and it's doing the pathway we just talked about, wouldn't it be remarkable if we went ahead and we know we're going to start with enzyme A, right? So here's A, and enzyme A was localized in the membrane around where we needed the product we were making. So let's think about what else. Now I'm not doing tails of any kind. Don't think I'm trying to make a point of saturated or unsaturated. I'm not. So once A's done, we have 2. B is going to act on 2. Wouldn't it be nice if we localized, knew where to find enzyme B? It's right there next to A in the membrane where they can become part of the pathway. Keep going. You know what I'm doing. There I'm going. So B acts on 2, right? 2 came from 1. So why don't we do that while we're here? A is going to convert 1 to 2. 2 is right here. How convenient. Because B converts 2 to 3. Well, what acts on 3? C. It would be really convenient if we localized C right here in the membrane so that 3, because it's right there, would be able to be acted upon by C to create 4. And you gotta love it when you know the end of the story. D acts on 4. It would be really convenient if D was right there localized in the membrane where we were able to convert 4 to 5. Now, I'm not trying to act like that these are literally in order like a true assembly line, but localization means in proximity to each other. So they're close, they're localized in the membrane of where we would use the product. So this is going to be the membrane of wherever product 5 is used. We make it where we use it. And we put the enzymes together so that these reactant product, because one is the reactant for A, the substrate, and it makes 2 as a product. Well, B uses 2 as a substrate and makes 3. So each one of these, the product of the first one is the substrate of the next one. So this is an assembly line. All it is. And it's going to maximize efficiency because you're able to produce your product where you're going to use it and you aren't hunting for something. So if we got, let's say, this far and we had made 4 and we had anybody seen D? Hey, where's D? We need to make 5. Oh, I hadn't seen D. I don't know where D is. Well, the whole thing's done. Like, you're done. So you've got to have your ducks in a row. Now you know where this comes from. I don't know where this comes from, but it sounds like a good one. You've got to have your enzymes in a row, localized. So localization of enzymes, all that means is you want to have all the enzymes for one pathway in the same location. Luckily, you can lock them into the membrane. Now, they're going to float some along with the molecular movement of these phospholipids, but in general, they're going to stay in proximity to each other, and I bet we don't just have one assembly line. I'm pretty sure we have a bunch of them. But they're all going to be right there. There's no hunting for D because we don't know where he went. Everything you need is right there. It is highly efficient. So we'll put therefore. This is highly efficient. Now, the other thing it does, going back to that feedback inhibition, if A is right here with B, C, and D, no problem as 5, the product, builds in concentration, there's going to have many, many collisions with A because it's right there. Easy to turn off. Most of the time, too, what you get is you get the ability to have specialized places where something occurs. So this pathway, if we only use the product in this location, you're not going to find these enzymes in membranes anyplace else. So an example of that would be the enzymes that are in that inner membrane of the mitochondria that do electron transport chain. So they're not going to be anyplace else. They're actually enzymatic pumps. But regardless, they're going to be this localized where we need them. And so it allows for specialization. So if you are in the cell and you're new and you're looking for 5, they're going to be able to tell you, hey, we only make that over there because that's where we use it, so go over there. And so you have these just specialized areas which lets you create the perfect microenvironment. So at that point, you're going to have the microenvironments because they're all doing the job that needs to be done. They're in the same spot. So these microenvironments exist that are going to optimize the conditions for these proteins. Remember what we talked about, all of this right here. You're going to be able to keep them at their optimal temperature, their optimal pH. All of those things you can do because we keep them all right here. We localize them. All right? So that's the end of that one. I think that's the end of the whole unit. So now what I'm trying to decide is if we're going to make study videos. I think I will, just not today. I've made many videos. So I will make you study videos if you felt like those helped. I don't mind doing it. But we're through with the unit. So we'll test here soon. I think you're wonderful. I can't think of any reason that, we'll have problems as far as scheduling, but you know how we are, we're flexible. So, all right, you're the best.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Our cells are amazing molecular machines. Every day, they convert food into energy, such as ATP, that drives the millions of biochemical processes necessary for keeping us alive. The pathways used to convert substrates, such as glucose, into products are collectively referred to as metabolic pathways. The drivers of these metabolic pathways are enzymes. Enzymes are usually proteins that work to assist chemical reactions by building or breaking down molecules. However, enzymes don't drive reactions at a constant rate. The rate of reaction can speed up, slow down, or stop completely according to the cell's needs. This process is analogous to an assembly line where raw materials are converted into products. The assembly line must be monitored to ensure that the supply of products does not exceed demand. If products are being created at a rate that is faster than they can be used, both resources and products are being wasted. The assembly line can produce at a slower rate or stop completely to prevent waste. In cells, this process is called feedback inhibition. Feedback inhibition is achieved through a process called allosteric regulation and plays an important role in many metabolic pathways. Enzymes have a unique area called the active site where substrates can bind. Enzymes that can be regulated have an additional area called the allosteric site. Downstream products can bind to this regulatory site, causing the enzyme to change shape and prevent it from binding with its substrates. This causes the entire pathway to halt, and new products are no longer formed. Conversely, when the amount of downstream product gets too low, the enzymes interact with their usual substrates and begin forming products again. Feedback inhibition plays a critical role in preventing cells from wasting resources and energy. Mutated enzymes that do not respond to allosteric regulation have been linked to disease states such as cancer. Many processes in our bodies rely on feedback inhibition to keep everything running smoothly and efficiently, maintaining homeostasis.